Proximity ligation assay allows users to detect protein-protein interactions in situ, revealing spatial and temporal patterns of the interactions. This technique can help answer important questions about regulation of C. elegans development. This technique offers comparable sensitivity for detection of protein-protein interactions without the complexity of other techniques such as FRET and BIFC.
Individuals who have never dissected worms to release the gonads may struggle with this step. Practice dissecting worms and use them for immunofluorescence to perfect the handling and dissection techniques before moving on to PLA.
To begin, pick 30 to 40 young adult worms into a watch glass dish containing 500 microliters of 1,X-M9 and levamisole. After collecting the worms, carefully remove and discard most of the media to remove bacteria that is transferred along with the worms. Add in fresh 500 microliters of 1,X-M9 and levamisole, and use the pipette to gently draw up and dispense the media to rinse the worms. Carefully remove and discard most of the media. Repeat the wash 2 to 3 times. After the washes, leave the worms in about 100 microliters of media for no longer than 8 minutes. Using a glass or polyethylene pipette, transfer the worms to a 25 mm by 75 mm microscope slide. Coat it with 0.001% polyl-lysine. Remove the excess media so that approximately 15 microliters of media remains on the slide.
Removal of excess buffer is critical to ensuring that the dissected worms and gonads will adhere to the slide.
Under a dissecting microscope,